Hi everybody. So this is chapter 6, a few slides on mitochondria. We'll do more about mitochondria in chapter 9. So ATP is produced via the mitochondria, and the vast majority of our ATP is produced by the mitochondria using aerobic cellular respiration. And if I asked you, just for fun, think of a typical cell and you had to count how many ATPs a typical cell would use in a second. Just take a guess. How many? What do you figure? About 1 billion ATPs per second. Try and think of how many anything we can do 1 billion times per second. Well, I guess we can make ATP 1 billion times per second. So with the architecture of mitochondria, it is bound by two membranes. So you have your outer membrane and an inner membrane, and notice that the inner membrane, the surface area is increased by folding in and out and in and out. Okay, so increased surface area. You can just think of it, imagine if you're drawing a circle, the amount of chalk that it would take to draw a circle versus the amount of chalk that would take to draw a circle but invaginated in and out and in and out. That extra surface area tells you something about the importance of the inner membrane. Must have something to do with a important set of enzymes that catalyze reactions that you need to maximize the amount of membrane so you can maximize the amount of the enzymes and proteins that are embedded in the membrane. And you would be right. The inner membrane, it has a lot of the cellular machinery for generating ATP. And so they have protein complexes that we can maximize the amount of our protein complexes by increasing the surface area without increasing the volume. So, and we'll talk about that more soon. So these invaginations are called cristae, and they can increase the surface area 4 to 5 times more than the outer membrane. Okay? So just by invaginating, you can get a much higher amount of surface area compared to the outer membrane that isn't invaginated. So remember that figure of an animal cell where it has a few mitochondria, a nucleus, one nucleolus, maybe a little clumping of Golgi. And I said that this isn't an actual cell, it's just showing a little bit of each organelle. But in cells that require a lot of energy, you're going to see, as you would expect, a lot of mitochondria in those cells of ours. So depending on the cell and its energy needs, you can get hundreds of thousands of mitochondria. And so our skin, not many mitochondria relatively, because our skin cells don't have a whole lot of metabolic activity. But our heart cells that need a lot of energy to beat, they'll have a significantly greater amount of mitochondria. And this is a beautiful slide that shows the cell, which is outlined here, and it's actually showing the mitochondria in red. So this cell has been stained, and the red dye is associated with the mitochondria, so you can actually see the location of the mitochondria. So our genome consists of the DNA that's in our nucleus, which are linear chromosomes, and that's where the majority of our genome is located, in our nucleus. But mitochondria have their own DNA. It's not linear, it's actually circular. And because the cell has so many mitochondria, and because the mitochondria can have a lot of copies of this circular DNA per mitochondria, a lot of times when the when you're doing forensics and trying to get DNA evidence, if the DNA is poorly has a lot of degradation, the sample is quite poor. One thing that they will do, instead of looking for the DNA that would have been in the nucleus, they look for the DNA in the mitochondria, because there's so many more mitochondria in a cell than there are nuclei. And in the mitochondria, there are so many copies of the mitochondrial genome in one per mitochondria. It can be a source of DNA for trying to identify individuals. It's because there's not as much, the genome of mitochondria is significantly smaller than the genome of in our nucleus. So because there's such a small genome in mitochondria, it makes limitations to how well we can identify an individual just based on mitochondrial DNA. So this cartoon is actually from a student, Carla, who thought I would like it, and yes, I did. If you ever find cartoons or pictures that are sciencey that you think I'd like, send them on. I would probably love them. And so this cartoon depicts the origin of mitochondria in the evolution of life on Earth. And there were some things about mitochondria that are interesting. One is that mitochondrial genome is circular rather than the linear chromosomes in our nucleus. And another thing is that the mitochondrial genome has the genes do not have introns. And another thing about the mitochondrial genome is that the genes of the mitochondria are very similar to the genes of bacteria. And it actually provides insight into the origin of mitochondria, because mitochondria have circular genomes, bacteria have circular genomes. Mitochondria have bacterial-like genes. Bacteria have bacterial-like genes. Bacteria don't have introns. Mitochondria don't have introns. And there's a theory, endosymbiotic theory, which is actually my second favorite theory, and it describes how the origins of the mitochondria as being early endosymbiotic events early in the history of life on Earth, and that the first eukaryotic organisms did not have mitochondria. But through a bunch of events where the ancient eukaryotic cell enveloped and enclosed an ancient prokaryotic cell, and normally that prokaryotic cell would be degraded by the eukaryotic cell and break it down for energy. And this is not one event, but many events over the early history of life on Earth. The mitochondria and the ancient prokaryotes weren't degraded and weren't digested by the ancient eukaryote, and they actually had a symbiotic relationship where the bacteria were able to provide food for the ancient eukaryotic cell, and the ancient eukaryotic cell provided a safe home for the ancient prokaryotic cell. And over time, the bacteria lost genes and became completely dependent on the ancient eukaryotic cell. It could no longer live without the ancient eukaryotic cell, and vice versa. So not all of the genes in the mitochondrial genome are enough to support the mitochondria. What that means is that some proteins that are needed in the mitochondria some proteins are actually encoded by genes that are in the nucleus. And those genes in the nucleus get transcribed, then get translated, and those proteins are targeted for the mitochondria. So, a little interesting history, ancient history. And like all our genomic DNA, it's double-stranded. Just like bacterial DNA. All cellular organisms, their genetic material is double-stranded DNA.